Welcome to EPA's free Small Systems Monthly Webinar Series, Challenges and Treatment Solutions for Small Drinking Water Systems, hosted by the Office of Research and Development and the Office of Water. A note, if you want to see the recordings of this webinar series of previous webinars, you can click the link that's on the top right of this screen on your slides that are in your handout section. Also, and you can reach the webinar series that way. Today's topic is disinfectants and disinfection byproducts. The slides for this month's talk, as it's going on, can be found in the handout section of the dialog box that pops up, so you can follow along with those slides if you wish. You can ask a question of the speakers, and we encourage you to write your questions in the question box in that dialog box, and go ahead and type them in while the speaker is presenting, and we can help organize them and get them ready for the question period, which will happen after all the speakers are done. Somewhere around 3 o'clock Eastern time is when we'll start the question and answer period. If you're interested in webinar support, if you're having problems with this webinar, the link is right there. Or you can send an email to that address and request support if you're having problems. And finally, if you're looking for a certificate of attendance for this webinar, it is available. The requirements of the certificate is that you must be registered or in a room with somebody who is. You must attend the webinar for 60 minutes. And if you're in a room with others, the name of the people that are not logged in must be provided by the person who is. So all certificates will be sent to the email of the person that's logged in. And now this is a little bit of a change here. Is in the past, we always had you type in the question box how many people were in your room if you had more than one. We'd like to do it a little different way. So you can either click on the link that's in the chat section and input it through that link, or you can pull up the Adobe slides that are in the handout section, go to this slide and click on this link here on that slide, and you can get again to the place where you can log in how many people are in your room. And only do that if you have more than just yourself that are logged in. And we recognize that this is maybe just a few extra clicks on your part, but it's really important to us to help justify the expense of the webinar series by accurately recording how many people attend. So we really appreciate you going through that and clicking on that link and telling us how many people are in your room if there are more than one. And please be safe. A couple upcoming webinars I just wanted to plug. October 28th, health effects associated with harmful algal blooms and algal toxins. November 18th, drinking water models and tools. And November 17th, creating resilient water utilities. So please put these on your calendar. You can, again, go to these slides and click on these links if you want to register or looking for additional information on these webinar series. Please put them on your calendar, and we'd love for you to attend these webinars. Okay, with those housekeeping duties finished, I'm going to now introduce our first speaker, Dr. David Wahman. Dave is a environmental engineer with the EPA's Office of Research and Development, or CSER, the Center for Environmental Solutions and Emergency Response, where he focuses his research on disinfectant chemistry, disinfectant biofilm penetration, and distribution system water quality issues, including nitrification. He's a registered professional engineer with over 24 years of experience, with a BS degree from Rose-Hulman Institute of Technology, and a master's and PhD from the University of Texas at Austin. And with that, I will hand the podium to Dave, who's going to present our first presentation today.
Okay, thank you, Tom. I'm assuming everybody can hear me.
Yes,
first thing I want to do first thing I want to do is acknowledge my colleague on this work, Jonathan Pressman. This is an area that we've been working on for about the past 12 years or so, looking at how disinfectants penetrate biofilm and more recently sediment. And a couple things I want you to walk away with after this webinar. I'm going to talk about two general areas of some research we've done. And one is the aspect of how disinfectants have penetrated nitrifying biofilm. We're going to look at both monochloramine and free chlorine, how that occurred, and also not look at how it just penetrated the biofilm, but then what did it do when it got there? Did it kill the bacteria? So we'll look at some viability on that. And we'll also look comparing how the disinfectant penetrated that biofilm on a one-dimensional basis and also in a three-dimensional basis to see if there's any impact to heterogeneity of the biofilm. And the other main area that I'm going to talk about is the storage tank sediments. So this is sediment we actually got out of the distribution system. And we also kind of repeated some of these experiments looking at, again, monochloramine and free chlorine, how they penetrated storage tank sediment. Also looking at not only viability, but now we're looking at activity. So how active the biofilm are, as well as we also looked at the impact of disinfectant switching, or more commonly characterized as a chlorine burn for systems that are chloramine systems, where they switch from monochloramine to free chlorine and back to monochloramine. So our work on this started really looking at nitrification in drinking water. And then if you're unfamiliar with nitrification in drinking water, it occurs because we have ammonia in the water. There's bacteria in there that'll readily oxidize that ammonia. And you get ammonia in the water from a couple ways. Well, when we use chloramines, we're adding ammonia and free chlorine together to form chloramines. So we can have some ammonia left over from that process. Also, when chloramines then break down, they release ammonia back into the water. We can also have naturally occurring ammonia. So you can get ammonia in your water in a variety of ways with the chloramine system. If it's in the water, there's bacteria that will gladly oxidize. That's nitrite, and that's commonly been referred to as ammonia oxidizing bacteria in the past. More recently, we know there's another class, ammonia oxidizing archaea, who can also do this first step in nitrification. Once you have nitrite, there's another class of bacteria that will readily take that to nitrate if they're around. Those are nitrite oxidizing bacteria. And then within just the past few years, we kind of found out that you don't need both of these classes to even do this process. One bacteria itself, combined ammonia oxidizers, can actually do the process by themselves, and they don't need two classes of bacteria. So if you have ammonia in the water, you can have some biological activity, which you don't want. And basically, if you just think about nitrification, you have bacteria growing on ammonia, you have a disinfectant there and it's killing them. So for them to survive, they need to grow faster than they're getting inactivated. And part of understanding this kind of which side this tips the balance to is understanding the disinfectant concentration, and not just in the bulk water, but what's actually in the biofilm where these bacteria might be growing, or near pipe walls or in sediment. So that's kind of where our research kind of took off the last couple of years, because we didn't really have that characterized. We kind of felt like we understand like maybe monochloramine penetrates better, but we didn't really ever measure that. So we wanted to do that and then compare that to free chlorine. So the research we conducted, we actually measured disinfectant in biofilm and sediment. We did this over time, and we used microelectrodes to do this. And I'll explain what those are in a second, but mainly they are miniaturized probes. So we used that to measure the disinfectants. We also measured the viability of the biofilm after they had been exposed and the activity while they were being exposed. Again, we did this over time. For viability, we added a stain to the biofilm. Basically, you'll see some slides. If it's green, they're alive still or viable. If they're dead, that's an indicator that they're not viable. Or if they're red, and that means they're dead. And then we also looked at their activity of the bacteria. So looking at the important components of nitrification: dissolved oxygen, ammonia, nitrite, nitrate, and pH. And then we also, again, looked at disinfectant switching, going back from monochloramine to free chlorine, back to monochloramine, and see how that impacted how the disinfectant penetrated things. And then we finally looked at heterogeneity impacts of the biofilm. Was it good enough just to look in one dimension, or do we need to look at them in three dimensions to understand what was going on? So we published this literature over the years, within the last 10 years, in a number of publications. And these are just the headers of those publications if you want to refer to them after the talk to get some more information. But I'm just going to be summarizing some of this work today. So the first thing I want to do is introduce you to microelectrodes. Just think of them as very miniaturized probes of things you might find in a normal drinking water lab. They come in kind of two classes: an amperometric type, which is akin to a dissolved oxygen probe that you might have in a lab. This is where we apply a potential to the tip of the electrode. It causes a reaction to occur with the constituent of interest. and that generates a current, and that current is proportional to the concentration of what you're measuring. And this applies to like the DO, the oxidants, and then we also use this for nitrite. The other type is potentiometric, which is more like a pH probe in the lab, where you have a selective membrane and you develop a potential across it, and that potential that develops is then proportional to concentration. And what I'm showing here at the bottom is just a schematic of the 3-chlorine monochloramine microelectrodes. I'm not going to go into in great detail. The one thing I want to point out is that the tip diameter on these things is around 10 microns. So very, very small, very, very fragile too. And so the idea of the tip diameter, why it's important, is because that determines our resolution. So we get the resolution about twice the tip diameter. So if you have a tip diameter of say 10 microns, we can measure then every 20 microns in depth into a biofilm. And just to give you an idea of the scale there, depending on who you are, your human hair is about 100 microns thick, and your red blood cell diameter is on the order of 6 to 8. So we're more towards the red blood cell size on how we're making these probes and using them. So just to give you an idea of the scale here. This just gives you a picture on the right here of our microelectrode setup, and then at the bottom is kind of a mock-up of the flow cell that we use when we're actually doing the profiling, when we're using the microelectrodes on the biofilm. And basically we grow biofilms on a slide, lay the slides flat in a channel, flow liquid over them at a rate to keep the bulk water concentration constant, and then we profile, push the microelectrode down into the biofilm. And we use relatively thick biofilms so we can take some data. And we use a confocal scanning laser microscopy to look at the viability. And again, we did 1D and 3D profiles. So let me kind of bring this to like real life, what we're trying to look at here. So here's just a cross section of a pipe. It has a pipe wall, obviously. We have the bulk water flowing in the middle of it. And then around the outside, kind of the pink area here, I'm illustrating a biofilm. It might be growing attached to a pipe wall. And what we're trying to look at in this research is what's going on right where I put this black circle up. So right at the pipe wall, the biofilm, and the water near the biofilm. So if you blow that up, it kind of looks like this, where you have this water on the outside of a biofilm next to a pipe wall. And what we do essentially when we're doing our microelectrodes is we put a microelectrode in here, and it kind of sets a scale then. So we can show concentration on the y-axis, and then we'll move the microelectrode on the x-axis and take those concentrations. So we always define in the biofilm experiment the surface of the biofilm with a distance of 0. The distance is moving out into the bulk water as a negative distance, and then distance is moving into the biofilm as positive. So here we have about a 300 micron biofilm. So we take our microelectrode and we start moving it towards the biofilm and take measurements. The first area we're going to hit is the area that's essentially the bulk water. So this is on all these profiles, the bulk water kind of represents if you went out to a pipe in a system and took a water sample, what you would measure. It's the kind of the completely mixed water in the bulk phase. And we can see that as we're doing microelectrode profiles, it'll be this kind of horizontal constant concentration. When you get to within a couple hundred micron of the biofilm, though, you start to see an area that's represented by the liquid layer, or a stagnant layer where only diffusion is occurring. And diffusion is just things move from high concentration to low concentration. So that's why you see this kind of downward slope here. And the concentration is decreasing in this stagnant kind of liquid layer. And it's just moving here through diffusion. And then you enter the biofilm where the action is taking off. This is the area that's consuming things. And so you'd have both diffusion going on still in the biofilm, but now you have the reaction, which is what's causing the concentrations to decrease. So you kind of see these phases in these profiles just to keep an eye out for. And just to point out, the slope of this stagnant layer, this diffusion layer, kind of tells you something about what's going on in the biofilm. The steeper this is, the more of the chemicals being delivered to the biofilm, which kind of indicates that that's having a greater reactivity in the biofilm. So just keep that in mind as we're looking at these profiles. So I'm going to start with the first set of experimental data here. So we're feeding monochloramine, running that across the biofilm, and then we're going to take measurements over time to see how it starts penetrating the biofilm. So the first time point I'm showing here is 10 minutes. So this is the profile at 10 minutes. We just started flowing it. So it's just starting to increase into the biofilm. You can kind of see that flat area out in the bulk water, which is what we were feeding, about 2.5 milligrams per liter. And then the decrease to the liquid layer and into the biofilm. After about an hour, you start to see the concentrations coming up in the biofilm. So it's starting to penetrate the biofilm slowly, but it's getting there. 2 hours, it keeps moving up. 6 hours, we're starting to get a pretty decent penetration. And then finally 24 hours, we've reached a full penetration of the biofilm. So we have the concentration is the same as the bulk essentially. So with monochloramine over a period of somewhere between 6 and 24 hours, we got complete penetration, meaning we were able to diffuse into the biofilm faster than the biofilm could react away the monochloramine until we got kind of a complete penetration, which is what we expect with monochloramine. And just to point out, it took between 6 and 24 hours to reach this full penetration. If there hadn't been any reaction occurring in the biofilm and things were moving by just diffusion only, we should have gotten this horizontal line at about 10 minutes. So it's not the monochloramine isn't reacting, it's just that it's able to eventually react with the things it wants to and penetrate. Then we took a similar biofilm, we repeated the same thing with free chlorine, and you'll see what that looks like now. So we started feeding free chlorine at about 2.5 milligrams per liter, and after about a half an hour, we have very little in the biofilm and a very low surface concentration where the dashed vertical line is. At an hour and a half, looks exactly the same. It's just sitting there. It's not penetrating into the biofilm any more than it initially did. 8 hours, same thing. 20 hours, still not moving into the biofilm. So it's kind of reached kind of a pseudo-steady state here, where it's essentially can only diffuse to the biofilm as fast as it's being reacted away. So different from monochloramine, free chlorine stabilized without complete penetration. It was diffusion limited. We couldn't get enough chemical there to make it go in further, and it only penetrated the very surface, about 50 microns, or the very top layers of the biofilm. Much different than monochloramine. One thing you can do to kind of help it penetrate is you can increase the concentration. Again, to deliver more chemical to the biofilm. So here, I'm showing you, this is the initial penetration profile at 20 hours. Again, very little penetration. And that was about 2.5 milligrams per liter we were putting through the bulk water. We raised that up to about 10. And the first thing you're going to notice is we got a little further in. The slope is much steeper. We're delivering a lot more free chlorine to the biofilm here. And you're starting to penetrate it a little bit. After 15 hours, it really hasn't moved much yet. At 51 hours, though, we finally start seeing it starting to penetrate. So it's finally starting to burn through the biofilm with this much higher delivery. And then 63 hours, it's continuing to progress up in. So with lower concentration, we kind of stabilized without complete penetration. There's just so much to react with there, it couldn't really get in. As we increase the concentration, we could start moving towards full penetration, which is what we expected, but it's a high concentration. And again, if you were measuring the bulk water from a pipe, you might think you have 2.5 milligrams per liter in the bulk. You have that in the bulk water, but you're getting very little into the biofilm. Then we took a biofilm that we had completely exposed to monochloramine and then exposed it to free chlorine. So we kind of did the monochloramine and free chlorine back to back on the same biofilm. After 24 hours, we had completely penetrated the biofilm with monochloramine, the horizontal line I'm showing here. And then we started adding free chlorine to see what would happen in this case. As I went a little too fast. At 20 minutes, free chlorine starting to penetrate. 8 hours, it's kind of locked in there again. It's not going any further than what it did at 20 minutes, and maybe moving in very slowly at 24 hours. So this is a biofilm that has been completely penetrated with monochloramine. We add free chlorine and it doesn't penetrate hardly at all. So what this tells us is that, you know, monochloramine got the complete penetration. Free chlorine, it again stabilized at some low penetration into the biofilm. And then the monochloramine and free chlorine are obviously reacting with much different things in the biofilm, and much different rates. And monochloramine being a much weaker oxidant than free chlorine probably explains that. We also, at the end of these experiments, looked at the viability of the biofilm. So these are surface images from the three scenarios I just showed you: monochloramine only, free chlorine only, and the middle picture is monochloramine, then free chlorine. And the imaging technology we were doing here could only get to the very upper limits of the biofilm, maybe about 50 microns depth. Again, these are about 300 micron biofilms, so only the kind of upper surface. And you'll see that green here again indicates viable or live, red indicates non-viable or dead, and you can kind of see the differences with the exposures. So for monochloramine, when we use monochloramine only, even though it penetrated completely, we still see viable areas after 24 hours. Again, it's a weaker disinfectant. But for the free chlorine applications, both of them, we see completely red. So again, these are only looking at the top layers, which tells us that what free chlorine gets to, it's pretty well knocking it out. but it's a matter of it getting there. Like I said, that was all 1D. We were kind of going in kind of one or two places into the biofilm, and then we did more research after that, looking at a thicker biofilm, looking over an area of the biofilm to see if there was any real differences in terms of some heterogeneity or anything. So here we're looking over these are kind of profiles over a 400 by 400 micron grid, and these are kind of layers in that. And then you'll notice that all the sheets are kind of flat. So we weren't really seeing much heterogeneity here. So it was kind of like a homogeneous penetration on the scale we could measure. And overall with the 3D stuff, what we found is we saw similar trends as the 1D. Monochloramine penetrated faster. Free chlorine did eventually penetrate if we gave it enough time. And again, if we exposed the biofilm and completely to monochloramine first and then brought in free chlorine later, it was like the biofilm really never saw anything before the free chlorine. The free chlorine kind of do what it wanted to do without, with or without being exposed to monochloramine first. The one thing we did add in these experiments were we were able to get full depth viability measurements here. So these are the cross sections of the biofilm after 7, 10, or 16 days of monochloramine exposure. It's a little bit hard to see on your screen, but there are red and green areas here. And I just kind of want to summarize those with some graphics to kind of illustrate what we were seeing. And that basically initially, again, we got monochloramine penetrated. It just took a while to kill things. So you saw it being this mixture of green and red at 7 days, got a little more red at 10 days, and then after 16 days, it was kind of pretty much red throughout. So it just sits there and works and works. For the free chlorine, it's a little bit different. Again, cross sections of the biofilm anywhere from 2 hours up to 13 days. This you'll notice a little bit different if you can kind of pick it out. Again, I'll summarize this in a second, but kind of more like a red defined area and a green defined area. And so what we were seeing kind of here was this defined line again in a narrow range between viable and non-viable. Basically where chlorine got to, it was turning it red. The other thing we saw over time on this was actually we're starting to slough the biofilm. So we were decreasing the thickness of the biofilm over time. So at 13 days, we had a much thinner biofilm that ended up there than what we started with. This is a picture out of one of our manuscripts that kind of just summarizes the biofilm summary and the idea that monochloramine kind of can penetrate things and then slowly work on it, whereas free chlorine kind of eats it from the outside in, was kind of what our experiments were showing. So that kind of talks about the nitrifying biofilm work, which is the majority of the work. And then more recently, we've done some work with storage tank sediment. I want to get into that here for the remainder of my time. So with the sediment, we got it from distribution system storage tank. We put about 20 centimeters of the sediment into a Teflon cup, put about 5 centimeters of water over it, and changed out that water real quick so we maintained a constant concentration of 4 milligrams per liter of either free chlorine or monochloramine, depending on what we were feeding over the entire study. And we exposed this sediment to essentially 120 days of monochloramine, followed by 60 days of free chlorine and 60 days of monochloramine. Just to give you an idea how this one worked, so here's a schematic of that. We got the Teflon cup, the sediment at the bottom, and the water at the top. Just think about this kind of flipped on its side. We have a water area, sediment area. Again, concentration on the y-axis now, distance on the x-axis. 0 is going to be the sediment surface. Negative out into what would be the bulk water, what you would measure taking a sample in a system. And then positive distances into the sediment. And here we're actually measuring just the pore water in the sediment. And so we're going to move that in and get another profile like I was kind of showing you before. So in phase 1, we looked at monochloramine exposure. I'm just going to go through the monochloramine time profiles in time here. Reminder, our sediment was 20,000 microns deep or 20 centimeters. So you can kind of get an idea of how far the monochloramine is going to get in because I'm only showing you half the sediment here. So after 5 hours, we're feeding again 4 milligrams per liter in the bulk. We get a little bit of penetration into the sediment. 6 days, continuing to move into the pore water. A month, a little over 2 months, and then the final of that phase at 119 days. So in the sediment, the monochloramine did slowly progress inward, but we didn't come near close to getting all the way in. And just again, point of reference, if there was only diffusion occurring here to fill up the sediment with this depth, should only have taken about 2 weeks. So we got about 30% penetration with the monochloramine after 120 days into this sediment. So not too great. Then at the end, I'm just showing you all the profiles of what we did. We looked at a bunch of activity measurements here too. So this is a summary profile at the end where I'm showing dissolved oxygen, monochloramine, ammonia, nitrate. What you can see is the dissolved oxygen being the green line. You can see that decreasing in the sediment. The monochloramine is the profile I showed you before where that's going down in the sediment. You'll see that the ammonia line actually has a hump in it. And that's indicative of something that's being released from the monochloramine going away and then being consumed by the bacteria in there. And then that's turning the ammonia, converting it all the way to nitrate. As you see the nitrate or the light blue line increasing as you increase in depth in the sediment. There's a lot going on here, but it just shows us that nitrification is occurring. And the oxygen consumption kind of parallel with what the nitrification we were seeing. Then we moved on to phase 2, where we exposed the free chlorine. So here's the line from the end of phase 1, which shows you how far monochloramine got in. Then we started feeding free chlorine. 5 hours, really not in there at all. 1 month, we start getting a little penetration. At 60 days, it really hasn't moved from the 1 month period. So again, free chlorine hardly got into this at all compared to the monochloramine, only about a 3% penetration. So only the first half a centimeter. Or first 500 microns. So it barely penetrated at all. Again, much more reactive, not penetrating. At the end of the phase, here's all the profiles I showed you again. Again, dissolved oxygen still going down. Remember, now we're doing free chlorine. So we've removed the ammonia from the feed source, but we're still, even after 60 days, measuring nitrogen products in the sediment, which was quite interesting, to be honest, because we expect that to kind of flush out in the first couple weeks. Without it being fed in. So there's probably some cycling going on here in the sediment between the ammonia and nitrate. So it's still hanging around. So again, you know, in a real system, you switch over to free chlorine thinking you've gotten all the ammonia out of the system, we're going to shut down nitrification, but if you've got some sediment in there at the lower levels of that sediment, it still might be going on. And then finally, we went back to monochloramine for a couple months. So again, a reminder, where we ended phase 1, where monochloramine got, where free chlorine got at the end of phase 2, and we're starting to feed monochloramine again. So 9 days in, 22 days in, 43 days in, and 55 days in. So monochloramine is starting to go back to where it was at phase 1's end. Maybe penetrating a little bit greater now. Got about 40% penetration where we could actually measure some monochloramine, but still not anywhere near completely getting into the sediment after this time frame. Again, the final profile kind of went back to the end of phase 1. Dissolved oxygen going down, the ammonia hump for releasing and consuming it, and nitrates being produced. Again, I didn't mention this, but we did measure pH and nitrate in these experiments. pH, we had buffered the water, so it was pretty stable. Nitrite, we didn't really pick up that much. So it was really complete nitrification was occurring in the sediment. Just a quick summary on the sediment. We never got complete penetration. Monochloramine penetrated further than free chlorine. But qualitatively, we're similar to what we saw with the nitrifying biofilm in terms of the two oxidants here. One thing that was remaining was definitely biological activity indications that remained throughout the entire study. We mainly got complete nitrification. The oxygen was fully consumed in the sediment. So we go through the whole oxygen kind of conditions where you have aerobic at the surface, towards the surface, upper part of the sediment, go to anoxic about midway through the sediment, and then we would have had anaerobic at the bottom of the sediment. The disinfection switching, again, we still had measurable nitrogen species in the sediment the entire time we were on the free chlorine, and we did get nitrification to resume right when we switched back to monochloramine. Again, bottom line, you have to take nothing else away from this. It's a good idea to get the sediments out of your system. That covers the the two things I want to talk about today, the nitrifying biofilm work we did, the storage tank sediment work we did. And the final thing I want to do is acknowledge some people who helped us create this information. Paul Bishop has got us started with microelectrodes. We worked with his PhD student originally. His last PhD student, who ended up being a postdoc for us, is now at Central Florida. Woo Young Lee did all the nitrifying biofilm work, and then Hong Lu did the sediment work. And with that, we can go on to the next talk.
So I'd like to introduce, thank you, Dave, for the presentation, a great presentation on disinfectants. So our next speakers, there will be two, James Brown and Jose Rodriguez. James, or Jim, manages EPA's Safe Drinking Water branch in Region 6. During his more than 35 years of experience at EPA, He worked at the Resource Conservation and Recovery Act program on groundwater monitoring, site characterization methods, and statistical analysis of water quality data, and in the Office of International Affairs, where he co-managed an environmental improvements project in the Ural Mountains in the region of in Russia. Jim has a BS from Indiana University and an MS from Virginia Tech. And Jose Rodriguez, Jose is an environmental engineer in Region 6 Drinking Water Section. During his more than 35 years with the EPA, he's worked in the Drinking Water Enforcement Program at the U.S.-Mexico Border Infrastructures Program before returning to the Drinking Water Section in 2013. Jose has a BS from Texas A&M University, now Texas A&M. So with that, I will hand it over to you, James, for the second presentation.
Thank you, Tom. I'll wait for the slides to come up here.
Oh, and I should mention to the people on the list, if you haven't put in how many people are in your room, if you have more than one and you haven't used that link, you can go to the chat line and go to that link, or pull up the slides within the Adobe slides and go to the link that's, I believe, on the second slide. And that's the way we're trying to capture how many people are in the room. So that would be great. So with that, I'll hand it off to you.
Thanks much. Well, we're going to pivot from the laboratory to the field, as I think most of the viewers are probably very interested in how all this applies to ensuring the drinking water is safe and how we can do our jobs a little bit better in meeting MCLs. So today we're going to talk about a disinfection byproduct compliance improvements project that we have ongoing in Oklahoma. And we're really kind of midway through the project, but it's an important project in light of some of our new performance measures that we've been working on at EPA. And we wanted to alert everyone of the progress that we're making and hopefully come back in about 6 months and give a little bit more on the project findings. So this is being done by EPA Region 6 in Dallas, Texas, as well as the Oklahoma Department of Environmental Quality in Oklahoma City, and they have many field engineers that we're working with. But I also have to provide acknowledgments to the Office of Water, in particular Jennifer McLean, Anita Tompkins, Maria Carbo Lopez, Kathy Davis, and Ron Bergman, who all provided us with some funding and encouragement to go forward with this project. And also our colleagues at the Cincinnati laboratory and the Technical Services Center. I want to give recognition to Rick Lieberman and his staff, Allison Dugan and Matt Alexander. So a big shout out and thanks to everybody who's helping us with this. And then we also on our staff had Nana Edmond, who's working with both Jose and I, and the rest of the section, frankly, to help implement this project. So if I could have the next slide, please. So back in late 2017, the EPA came out with a new strategic plan that contained a transformational breakthrough measure. And that was to reduce the number of community water systems that are out of compliance with health-based standards by 25% by September 30th, 2022. And we are now midway through that time period. And we're making some progress, and that's what we're going to focus on today. I know that many of you who are out there working in the regional offices are working on this measure. And also a lot of states have developed similar measures, and they're helping us to reduce non-compliance at community water systems. Now, what's noteworthy about this measure is the focus on community water systems. In the past, our focus was on percentage of population served that met health-based standards. And what that did was it basically masked the small systems. And since this is more of a small systems webinar, you can see the dramatic impact. The EPA Region 6, we had about a 94% population that received safe drinking water that met all of our health-based standards, working under that metric. When we shift from a population-based to a community water system-based, you can see that Region 6 doesn't look so good all of a sudden. We have 1,072 systems that are out of compliance at a snapshot that was taken in the third quarter of 2017, which under our lean management system is called our jumping off point. And you can see that we're about 4 to 5 times higher than all of the other EPA regions. And there's several good reasons for that I'll go into in this talk, but you know, it looks like we're in first place and we are for non-compliance, and that's not a real comfortable thing or place to be in when, you know, you're being scrutinized at business reviews every month as a result of the lean management system. So let's go on to the next slide. So, you know, when you're faced with a huge non-compliance number, you start to ask yourself, well, you know, where are we falling short? We regulate over 100 different contaminants in drinking water, and we can't simultaneously try to improve compliance across all rule suites. Which ones do we really need to focus on? And you'll see in this graph, we did a pretty deep dive in our safe drinking water information system to see where the violations were. And the stage 2 disinfection byproduct rule stood out, and in particular in Oklahoma, Louisiana, and Texas. And it wasn't really surprising to us when we looked at the geophysical provinces that those states straddled. They're very, very thick organic soils called mollisols that have a lot of organic carbon. And in particular, in Oklahoma, they have a lot of drinking water systems, even the small systems that use surface water. So we have a lot of DOC in our natural source water. We're at the bottom of the Mississippi River watershed, of course. So, you know, we get a lot of dissolved organic carbon in our water from many, many other states. In fact, about 34 or 35 that drain into the Mississippi River water basin. And, you know, as a result, when we add chlorine to our water, and we have to have more than a trace in Region 6 because it's warm, we're struggling with issues in Louisiana and Oklahoma with Naegleria fowleri, and really a trace disinfectant just won't cut it. We have to at least have 0.2, and in Oklahoma, in fact, and Louisiana, they strive for 0.5 to protect against Naegleria and other microbial pathogens. So we're really, you know, our backs are up against the wall with that dilemma of how do we ensure that in the short term that any type of acute effects are safeguarded against through our disinfection processes in drinking water. And then we're balancing that against the chronic effects of forming these disinfection byproducts. So is there basically a way you can have your cake and eat it too in a simple parlance for environmental engineers? And that's what we're trying to decide. And that's why we have this research project going on. We also have another project with the state of New Mexico where we're looking at the groundwater rule violations, and we're trying to stem some of our less egregious significant deficiencies that we assign to water systems when we do sanitary surveys to try to help reduce those numbers. But for right now, we decided to go after disinfection byproducts rule and to partner with Oklahoma in this pilot project because they have a great area-wide optimization team that we've been working with for a while. And they really had the right kind of the right mindset. You know, when we approached our states with this, and states, they've been wrestling with recalcitrant systems for a long time, and some that are basically just intractable. So, you know, when you look at non-compliance in a region, certainly we have our own tribal program, and you know, you can kind of adopt the attitude that, you know, it's a money problem. The only thing it's going to take is a huge influx of money for these systems to add new infrastructure, get better operators, and then we'll see better compliance. On the other side of the spectrum, you have the attitude that, well, you know, some of them are just barely above the MCL, and maybe with some better operations and maintenance training, we can get these systems back into compliance. And we saw that attitude in Oklahoma, and they set up their SRF to have loan forgiveness programs and grants so that if there were minor infrastructure improvements needed at some of these small water systems, they could help facilitate that. So it was a natural pairing between Region 6 and Oklahoma to try to launch this pilot project. So the next slide, please. So this is a little bit about our lean management system that we're operating in. I know some of the states are starting to adopt that. Oklahoma is, Texas is, and Region 6. But the idea is to reduce noncompliance by 25%. And we are working with, the goal was to work with 20 different community water systems that had cooperative operators who would be receptive to the training and who would also adopt a neighboring water system who wasn't part of this study, who was struggling maybe with the same compliance issues, and help train them. So there's a pay it forward component of this that we built into the project. So we ended up with about 31 different water systems who signed up. They were all very enthused. And of course, we're ready to do these trainings and benchmarking, and then COVID happens. And we decided to try to continue with the operators who were willing to wear masks and do as much remotely as we could, because we really thought, based on kids that are being homeschooled and college students, nephews, nieces that we all have that are now doing their schooling remotely, that this may be part of a new normal. So we'd better get used to it, and this would be a good opportunity to see how flexible and effective these type of distance learning strategies are. So let's go to the next slide. We're trying to reduce non-compliance with stage 2 DBP. And we're going to talk a little bit about why our numbers are so high in Region 6. And I already mentioned the high content of organic matter in the soil, and state rules requiring 0.5 milligrams per liter of chlorine at the point of entry in at least Louisiana and Oklahoma, and our other states require at least 0.2. We have long distribution systems in Texas and Oklahoma and our other states. And with that, you've got higher residence times and more time for disinfection byproducts to form. We have warmer temperatures, which accelerate those reaction rates. We have a lot of consecutive systems, especially in rural areas of the state, which is why we partnered with some systems that have a wholesaler and a lot of retailers that Jose is going to talk about in a little bit. And you just, with all those factors, you end up with high concentrations of DBPs, and it's hard to meet the MCLs for trihalomethanes and haloacetic acids. So let's go on to the next. We thought what we would do is, in asking ourselves the question, well, what can we do about this problem? We're decentralized from the operator. We're in, our offices in Dallas, and we're not really the people that are turning the chemical feed valves at the chlorine tank. What really can we do? So we have a very mature optimization program in Region 6, and we actually partnered with Region 7. You can see in the light tan map at the bottom of the United States where Region 6 is showed and Region 7. And we thought, well, let's leverage our field experience in the optimization program and design training modules and do some diagnostic testing at these water systems to try to help operators on the ground with good practical management techniques to try to control DBP formation. And because we're also in the AWOT program with Region 7, we had, I convinced Mary Mindrup, and she gladly accepted my counterpart in Region 7, to do a similar study, which is ongoing right now. In fact, they're in the field. Allison Dugan and Matt Alexander are taking samples out in Kansas right now. So we'll have 2 different sets of results when we're able to come back and report to you on how we improved water system compliance by these management and operation strategies in both Oklahoma, as we're talking about today, and in the state of Kansas. So next slide. And I think I mentioned most of the study objectives here. We want to focus on operation and maintenance improvements first, rather than expensive infrastructure investments. If we could provide training and the best technical assistance that we can leverage as an agency to these operators, just how much of an impact, how much of an improvement in compliance can we expect? And that'll help, inform the 25% goal. Is it too aggressive, or is it, perhaps too lax? And this study will help answer that question. So we have water systems identified. We're looking at treatment, distribution, and tank management, and wholesale and consecutive systems to highlight some of the problematic areas that we've seen when we did a deep dive in our compliance assessment with CIDWIS. So that's the project scope. I'm going to turn it over to Jose now, and he's going to talk about some of the preliminary results and some of the takeaways that we've had so far.
Thank you, Jim. Let me advance the slide here. So again, this is Jose Rodriguez. I'm with EPA Region 6 in the Drinking Water Program, and have been working with this project since it started. So when we started working on this project with Oklahoma, the Oklahoma Department of Environmental Quality, ODQ staff, started evaluating and looking at historical data that they have for the community water systems with disinfection byproduct rule violations. And what they were looking for was a geographical area where they had a cluster of water systems that had DBP violations, and so they could reach the majority or a lot of the water systems and make it easier for them to participate in the training. So the initial list that they looked at consisted of 78 community water systems, which consisted of 13 wholesale systems and 65 consecutive systems. So what they wanted to do was, as Jim mentioned, look at wholesale or retail system and consecutive systems, and what the term they call the well, anyway, they wanted to look at those water systems and see how the wholesale systems might be affecting the disinfection byproduct at consecutive water systems. After many discussions between the partners, ODQ, EPA Region 6, EPA Technical Support Center, and our contractor process applications, that list was narrowed down to 32 water systems, 4 of those which are wholesale water systems, and the others are 28 consecutive systems. So if we look at this map here, this area right here is Tulsa, Oklahoma, and the systems that were selected to work on were the city of Nowata right here and the city of Veneta, which are just northeast of Tulsa, and the other area is southeast of Tulsa, which is the city of Muskogee and the city of Chakotah and their consecutive water systems. So out of those 32 systems, 31 of them agreed to participate in the project. The only one that did not was because the operator was retiring in July, which is about the time that the project was scheduled to start. So he saw it better not to participate in the project. So the training, the water operators would receive training, specialized training, very specific to their water systems on disinfection compliance training focused on disinfection formation and optimization and DBP control strategies. As Jim mentioned, we hope that some of these operators will partner with local water systems and pass on some of the experience and knowledge that they gain from this training. So I'll just show one of the combined distribution system for Muskogee. Muskogee sells water to nine water systems, and in turn the city of Haskell and the city of Porter sell to additional water systems. And the city of Muskogee is right in this area, so the consecutive systems are about anywhere between a mile 2 miles here in Muskogee 9 and Muskogee 6, all the way up to Haskell, which is 18 miles. And if you look at the additional distribution line from Haskell to Boynton, that's another 12 miles. So you're looking at, and these are just straight measured off of Google. You're not looking at the miles of mains that are involved, which are probably at a lot of distance for these miles. So you're looking at about 30 miles from Muskogee to Boynton, and then the other one that sells water is Porter and OK PWA. So that's another about 25 miles. So we do have long distribution lines that we have to deal with. So the training involves two separate training sessions. Each session will be offered in 2 locations. The first one is Nowata, and will serve the Nowata Veneta combined distribution system area. And the other one is the Muskogee Chakota combined distribution system area, and that one was held in Muskogee. The training is a 1.5-day training sessions. And the first one was scheduled to be done in July, but because as Jim mentioned, COVID came in and messed up our schedule. But it was eventually postponed and scheduled for August 17th, and we were able to have it on August 17th. And that focused on disinfection byproduct formation and optimization. I'll just go very briefly in a couple of the presentations that were provided at that time. The first one was an overview of the disinfection byproducts and their formation and why compliance is important. The other one was assessing DBP performance, and by that what we mean is ways of determining how DBPs are being formed in the distribution system, and other measures or other parameters that could be measured to help identify DBP problems in the distribution system. As you all know, analyzing for DBPs is expensive, so we're looking at other parameters that could be identified. And one that we used and one that we focused on was chlorine residual. There is some correlation with the lower the chlorine residual, there's a correlation that the longer the water age in the distribution system, and there's also that correlation of longer the water age, the more waters in the distribution systems, the higher the THM. So if we can identify areas where there's low chlorine residual, hopefully we can they will correlate to higher areas of DBP, and water systems can focus on those areas and hopefully reduce the DBP formation in those areas. The training also involved workshops where the participants were divided into separate groups, and that photo, this photo here shows some of the workshops. You know, that they're required to take their chlorine residual test, required to take distribution maps, and they did some calculations out in training in the field there. So the first workshop was on a calculated flush time and chlorine testing. You know, most operators, when they take a chlorine residual sample, they might flush the lines. You know, the number that we often hear is 5 minutes, or until there's a change in temperature in the water. But depending on the distance of the service line where the sample tap is to the main line, that might be too long or it might be too short. So there was a workshop on how to calculate the time it would take to flush the line from the main to the sample point. And then there's a system description and selective investigative sample sites. This is where the maps that the distribution maps that the operators took were used. The trainers sat with the operators and identified locations in their distribution system that they normally don't sample, but that might indicate problems with chlorine residuals or water age. So they identified those sites and then identified an investigative sampling plan that they were tasked with going in after the training and collecting samples at their distribution systems between the 2 training sessions. So the first training session took place. It was initially intended as a face-to-face training, but with COVID, the travel restrictions, and ODQ staff not being out to the field, it was changed to a virtual training, and then eventually it was decided to have a combined face-to-face training with virtual training, where the process applications staff went out and provided training, and we had a couple of ODQ staff also that participated in the training, and then the Allison Dugan from our technical support center provided some training remotely. So we were able to offer the training not only to people in the room, but also to others across the other operators, our Region 6 staff that wasn't able to attend, and other regional offices that we invited. We had about 30 staff from 5 regional offices that also participated in the training. The next training session will be November 13th, a week of November 9th through the 13th. This training session will be on DBP control strategies, and it will look at review some of the information that was provided initially, DBP formation, and looking at coordinated approaches to reduce those levels in the distribution system or DBPs. Also look at chlorine as a DBP indicator, as I discussed previously. They will also look at the operators were asked to not only collect samples, but record the data, have put it in a spreadsheet. So they will review that data during the second session and see how they can use that data to improve compliance out in the distribution system. Then what we want to focus on more at this time would be the in-plant or treatment plant and distribution focused control strategies. So we will look at various options for control strategies to improve compliance. The operators will then be tasked with identifying what would work best for their water system and implement it after the second round of training. And hopefully that will lead to what the compliance that we're looking for, without major infrastructure. Hopefully some O&M practices will help improve compliance. We did do some sampling initially, and this is a picture of a hydrant sampler that the Technical Support Center developed. It basically, if water systems are having trouble finding sites that they want to sample, or they know the areas long in the distribution system that have issues and don't have a sampling site, they can use this hydrant sampler, which is designed to reduce the flow to 20 gallons per minute once fully opened, and it does have a probe here for a temperature. For a thermometer, you can measure the temperature, and a valve here, we can adjust the flow to the stream that you want. So that is a tool that was described to the operators. So I'm not going to go through all the data that we have, because for one, we don't have it all, and we just don't have enough time to go through here. But I will go through the results for the Shakota sampling data. Up to date, we have done 2 rounds of sampling. One was done in July, prior to the training. We looked at the point of entry sites, and also the maximum residence time sites. As you know, the DBPs are required to be taken at specific points in the distribution system. One of them is the maximum residence time, and depending on the size and population, there might be other sites. But one site that is usually not sampled is the point of entry, and for the wholesale system, the point of entry is as the water leaves the plant. And for the consecutive water systems, the point of entry was identified as the meter where the water transfers from the wholesale system to the consecutive system. So if you look at the data for the first round that was done in July, there's some indication here that the DBPs are being formed. The MCL is 80, which is this red line, and it's very close at the Shakota as a wholesale system, very close to the MCL at this point. And as you go along the system, there's more DBPs forming out in the distribution system. So there's a possible option is to reduce the THMs in the plant, and hopefully reduce the formation out in the distribution system. So then we did another second round of sampling in September. The second round of sampling was done in September and finished in October. There were some issues with the sampling, but there is some improvement, and we at this point we can't put our fingers on what it is. You know, there's potential seasonal variations going from summer to fall. But if you notice here at Chakota, there was a drop from 69.970 to 56.9, which is a 19% drop in the DBPs at the point of entry, and not much. So there was actually a slight increase at the maximum residence time. But I will point out to a couple of other locations here in McIntosh 3. There was a big drop from 94. Wait a minute. Yeah, 94 at the point of entry to 60 down over here, which is a 36% decline in the THMs, and also in 5 McIntosh 5, it went down from 121 to 56, which is a 53% decline in the THMs, and also in 7 in number 7 there was a 27%. So there is some decline again. The operators were asked to take chlorine residuals, and some of the operators reported back that they actually took some chlorine residuals and did. And based on those results, if it was low, they started flushing the lines and getting fresher water out in the distribution system. So at this point, it's kind of difficult to determine whether that had an effect or it was more seasonal variations that we can expect. But we are seeing some drop in the DBPs at this point. Just yesterday, we received results for the Nowata and the Veneta combined distribution systems, but we haven't had a chance to tabulate them and do these graphs before. And so hopefully, like Jim mentioned, maybe in the future we can come back and present that data as well. This is the HA5 haloacetic acid, the 5 haloacetic acids. I'm not going to discuss much. There isn't much of a problem here in this water system. The MCL is 60, and they're very much below 30, 35 all the time, so they don't have that much of an issue with HA5s. We did take some chlorine residual samples, but again, there's not much variation. There's some, for some reason, there weren't some chlorine residual wasn't measured in the first round of sampling, and the second one there was a higher chlorine residual. Not sure if that's because the flushing that they did, but throughout the point of entry, the residual didn't change much. Next steps, we'll combine the results for the second round of sampling. The next training will be November, the week of November 9th through the 13th. And we do plan a third round of sampling, hopefully in January, when the water systems have already implemented some of their control strategies, and we'll have we'll look to see how effective that has been. Again, there is a seasonal variation there expected because of the colder temperatures in January. So we are looking at having another round of sampling, a fourth round of sampling, hopefully early to midsummer, to compare those temperature variations and also how effective the control strategies have been. The final outcome will be to have a draft and final report that is due to be completed by February. I know we won't have all the data at that point, but we will have a report on how we thought the project was, the effectiveness of the project. So in summary, we are working with, there are a lot of community water systems with health-based violations in Region 6 and Oklahoma, with the majority of them being DBP violations. We're working with Oklahoma on this project to identify methods of reducing the non-compliance. We have 31 water systems. In the first training session, we had 24 operators. I think the COVID-19 pandemic prevented some of the operators from participating in the training. We did have a few that participated remotely, so hopefully we can continue that participation in the second round of sampling. Some operators have conducted additional chlorine residual sampling and are implementing flushing programs at their water systems, so hopefully that will also help. Again, as mentioned, next training session is scheduled in November. And we'll continue to compile the additional sampling that's done after the data and compile and prepare those graphs. And we expect a report by February